Okay guys, in this video I want to discuss 3 different types of solutions and how those solutions influence osmosis, or really the diffusion of water across a cell membrane. So before we get started on this, make sure that you have a decent understanding of what osmosis is, how that works, what diffusion is, and the structure of the cell membrane, a little basics about that. Before I really go into these definitions, I also want you guys to have a decent idea of a couple of terms here. The first one is going to be solute. So anytime we talk about a solution, there's 2 words that you need to know. Solute is one of those. The solute is going to be whatever substance is dissolved into that solution. So if we look at the picture down here below, we have someone pouring a spoonful of, let's just go with sugar, into this cup of water. So we have a spoonful of sugar going in here, and it ultimately dissolves, and we have a nice what looks like just plain glass of water, but really it's a glass of sugar water. So the solute in this case is the sugar. The other word I want you to know is solvent. So let me spell that for you, because I hope you guys are writing this down. Solvent is spelled S-O-L-V-E-N-T. The solvent is whatever substance the solute is dissolved into. In this case, the solvent is water. Water is called the universal solvent because it will dissolve just about anything, except for lipids. So, knowing these 2 words is going to help us moving forward. The solute is what is dissolved. The solvent is what it is dissolved into. So, looking at the definitions of these 3 words, isotonic solution, hypotonic solution, and hypertonic solution. If we look at the definitions, an isotonic solution is a solution with an equal amount of solute as that inside the cell. So the solution is outside the cell. Inside the cell you have cytoplasm, which is water and a bunch of different dissolved particles, or a bunch of different solutes. An isotonic solution has the same concentration of dissolved particles as what you would find in that cytoplasm. So they're equal. This is also called equilibrium. The next word, hypotonic solution, is a solution with a lower concentration of solutes than what is inside the cell. If we break this word apart, hypo, this prefix here, hypo means less. Tonic is referring to the solutes, or the dissolved particles here. So this is a solution that would be outside the cell that has fewer dissolved particles than what you would find inside the cell. The last one is a hypertonic solution. Hyper, breaking this word down like we did before, this means more, and tonic again is going to refer to the solutes. So this is a solution with a higher concentration, or more solutes, than what you would find inside the cell. All 3 of these, influence osmosis, and osmosis is a form of passive transport. Going back to a real brief description of that, with our cell membrane, there are these proteins called aquaporins, and they allow water just to move back and forth in and out of the cell at all times in large amounts. And so water is constantly just freely moving in and out of the cell. If I place a cell in any one of these types of solutions, it's going to affect the way the water moves. So let's take a look at that. Alright, here's an illustration to help make sense of all of this. Let's start off here at the top with an isotonic solution. Remember, iso here is going to refer to the same or equal amounts of dissolved substances inside and outside the cell. So an isotonic solution, the solution outside the cell, in this illustration here, let's go with a 10% salt solution. And then inside the cell we have a 10% salt solution, or a 10% solution. So in this case, they're equal on both sides. Water's going to move in and out freely. Nothing happens to the cell. It's at equilibrium. This is what cells want. They want to have water going in and out at even rates, so nothing happens to them. Alright, this picture over here, if we look at this, the large pink circle is going to be our solute molecule. In this example, let's go with salt. And then the small gray one is going to be our free water molecule, which is our solvent. And remember, these can move easily across the cell membrane through the aquaporins, and so the cell's going to stay the same. Now, if we go to a hypotonic solution here, hypo means that there's fewer dissolved particles in the solution than there would be inside the cell's cytoplasm. So this picture here shows a 10% salt solution outside the cell with a 20% salt solution inside the cell. And this arrow shows that water is moving into the cell. So let's figure out why that happens. Over here, if we look at this picture, here's inside of our cell. It's got a higher concentration of dissolved particles and therefore fewer free water molecules here. Now outside the cell in the hypotonic solution, there's fewer dissolved particles, or at least a lower concentration of that, right? Well, if there's a lower concentration of dissolved substances, that means there's more free water molecules. So the water concentration gradient, the way the water wants to move, is from an area where there's more water, more free water, to an area where there's less free water. So the water is going to move into the cell, and it causes the cell to swell up. A hypotonic solution results in water moving into the cell. Now what happens here is it's only the water that's going to move. These solute particles are too large, or in the case of salt, it's a polar molecule, so it's not going to move freely across the cell membrane. The only thing moving here is the water. So water wants to even itself out. In this case, there's more free water outside here, and it's going to move into the cell. I'll explain that in a little more detail here in a second. Let's look at hypertonic next. A hypertonic solution means that the solution itself outside the cell has more dissolved parts, more dissolved molecules in it, so 20% salt solution, and then inside the cell we have a 10% salt solution. This is going to result in water moving out of the cell. Let's look at why that happens. Going back over here, we have inside of our cell a lot of free water molecules and a low concentration or a little bit of dissolved particles. In the picture that we have over on the left, it's the 10% salt solution. Whereas outside the cell, we have a 20% salt solution, which means that there's a whole lot more dissolved substance out here and fewer free water molecules. In this case, the water is going to want to even itself out, so it moves out of the cell. It's not going to move these dissolved particles or these solute molecules into the cell. They can't go across freely. They need a transport protein, possibly even energy. The cell might need to use energy to move these in. But water is just going to move either way. So the net movement, or the basic movement of water, is going to be outside of the cell. One way to remember this, guys, and this is where it gets a little bit backwards from your understanding of diffusion. Water is going to move towards the area that has more dissolved particles in it, because where there's more dissolved particles, there's less free water. So remember that water moves towards dissolved substances. If I have a 20% salt solution here, a 10% salt solution inside the cell, the water is going to move towards the more dissolved substances. And that's what this picture shows. The higher concentration of dissolved particles is out here, water's moving that way. Let's take a look at what effect this has on cells. So on this slide, I have the three different solutions, a hypotonic, isotonic, and hypertonic solution. The top row is going to be what happens to an animal cell in these solutions, and then down here we have the plant cells. So, in a hypotonic solution, water is going to move in because there's more dissolved particles in the cytoplasm, less solute concentration outside, which causes water to move into the cell. The cell swells up. I always remembered this when I was in school because hypo kind of sounds like hippo. Hippos are kind of a swollen animal, and the water moves in and it causes the cell to swell up like a hippo. And in the case of an animal cell, it could burst if it gets too large. That pressure could break the cell membrane and the cell dies. With a plant cell, because it's surrounded by a cell wall, it's actually protected from bursting like that. It builds up all of this, what we call turgor pressure, as that cell fills with water, and that cell becomes what we would say is turgid. A turgid plant cell has got too much water in it because it was placed in a hypotonic solution, because of that net movement of water in. With an isotonic solution, things are at equilibrium. There's an even amount of dissolved substances in and out of the cell, so water is just going to move in and out at equal concentrations both ways. The cell stays the same size. In an animal cell, this is what we would just call normal. In a plant cell, we refer to this as flaccid. So plants that have the right amount of water are just going to be looking normal. They're not wilted. They're not dying from having too much water in here and putting pressure on all of the organelles. This is what plants want. They want this equilibrium or isotonic solution around them. A hypertonic solution is where there is more solute, more dissolved particles outside the cell than what is inside the cell. So water, again, moving towards the dissolved substances, is going to leave the cell. In an animal cell, it causes it to shrivel up. Going back to the way I used to remember it, if it helps you, great. If not, I'm sorry. But I always thought of this as when I get hyper, I get dehydrated because I'm using all of my water. So hypotonic solution swells up like a hippo. Hypertonic solution gets dehydrated. In an animal cell, it shrivels up. If it shrivels up too much, then there's not enough water inside the cell. Things can't move across the cell. It causes the cell to die. When you drink too much soda, you've got all of this sugar water moving through your veins. And all of the sugar water has a higher concentration of dissolved particles out here. It pulls the water out of your cells and it dehydrates you. That's why drinking Cokes all the time will dehydrate you. In a plant cell, what we see down here is where the cell membrane is actually pulling away from the cell wall here. Remember, plant cells have a cell membrane inside the cell wall. When this happens, we refer to that as plasmolyzed. When something lyses, or this root word lyse, like we see up here, lysed, it breaks. So in this case, the plasma membrane, sometimes referred to as the plasma membrane, the cell membrane, is lysing or breaking away from the cell wall. So this is a plasmolyzed plant cell. This is what happens when you don't water your plants. The cell loses all of its water. Now, the cell wall does make the cell rigid. It gives it some structure. However, imagine there's hundreds of thousands of these little boxes stacked up on top of each other, making your plant stand up straight. Well, if I take all the stuff out of the boxes, those boxes are going to start to collapse on each other, and what happens then is the plant gets wilted. So when you start to plasmolyze your plant cells, they start to wilt. And this is what happens when a plant cell is placed in a hypertonic solution. It's why you don't water your plants with salt water. This is what kills plants when the storm surge brings all of the salt water from the ocean onto land after a major storm like a hurricane. And that water seeps down into the soil and it starts to pull the water away from the plant cells. So that salt water can kill plant cells. This last slide here is just it's a classic experiment showing the effects of osmosis and how this actually works. What I want you guys to see, though, is we have this U tube right here, called that because it looks like a U. And if we put a semi-permeable membrane that represents the cell membrane here, the only thing that's going to move freely across is water. These dissolved solute molecules are not going to go back and forth. Only water moves across, just like what would happen in the cell membrane. On this side, if we make a hypotonic solution, very few dissolved particles, just a little bit of salt or sugar, whatever it is you want to dissolve here. On this side, on the right-hand side here, we make a hypertonic solution, dissolve a lot of molecules in there. Our water, again, is going to move across because, remember, our membrane is selectively permeable, so the dissolved particles can't fit through there. They're not going to move. It's just going to be the water. And it pulls the water onto the hypertonic side. Just like if this was a hypertonic solution and this was inside the cell, water is going to leave the cell. So the hypertonic solution pulls the water over here, and what you end up with is this strange little phenomenon where you've got more water on this side than on this side. Because water always tries to even itself out, but in this case, because of the semi-permeable membrane, or the selectively permeable membrane here, it's only allowing water to go through, so the water itself evens out with the concentration of dissolved particles on either side. I hope this helps make some sense out of osmosis and how different types of solutions affect that. The one thing I want you guys to remember is water is always going to move across that membrane. You can't stop it. It's what's going to happen. The dissolved particles on either side stay there, and so the water then is going to move across that membrane and try and even itself out.